Hello friends, welcome to my channel Study Made Easy. I am Rushan Shah. In this channel, we cover all subjects, and if you are new on our channel, please like, subscribe and share it with your friends and press the bell icon so that you do not miss our other notifications. And we have made many previous videos about other subjects, so please do visit them. So today we are going to start on a fresh note with biology. We are going to take a new subject with a new chapter. So in biology, we are going to start with the chapter called Structure of Chromosomes, Cell Cycle and Cell Division. So in your previous classes, you must have heard the terms called chromosome, cell cycle, and cell division. So, what are chromosomes? So, chromosomes are highly coiled and condensed chromatin fibers. Highly coiled and condensed chromatin fibers. Now, understand the point. In biology, the same terms are very important if you want good marks. If you want to top in biology, you have to write same language, same words as and given from textbook. Don't try any other language. Not acceptable. For good marks, you have to write good words. So this is a definition also. They can ask you what are chromosomes. You have to write chromosomes are highly coiled and condensed chromatin fibers. They are made up of complex DNA and proteins called chromatin. Now, what is chromatin? Chromatin is a mixture of DNA and histones. These are molecules, DNA molecule and histones molecule. So, what is a macromolecule? A single DNA molecule is very large and hence is described as a macromolecule. The single DNA which is very large in structure is called as macromolecule. So DNA strands, what are DNA strands? They are called as nucleotides. Repeating nucleotides. Now what is nucleotides? Nucleotide is made up of three contents and these three contents together forms a nucleotide. Phosphate, sugar and nitrogenous base. These three together form a nucleotide. Phosphate, pentose sugar and nitrogenous base. Now, what are four types of nitrogenous base? Adenine, guanine, cytosine and thymine. Adenine, guanine, cytosine and thymine are the four nitrogenous base. So with this, we are clear with a DNA molecule. Don't worry, I'll also display a photo over here, or else you can also go to Google right now and you can see. Then histone proteins. After DNA comes histone molecule or histone proteins. What are these? They are proteins that help in coiling and packaging of DNA into structural units called nucleosomes. मतलब मैन्युफैक्चर होने के बाद जो पैकिंग होती है प्रोडक्ट की वो हिस्टोन प्रोटीन्स करते हैं और क्या आता है न्यूक्लियोसोम्स. देन अ सिंगल ह्यूमन, दिस इज अ वेरी नाइस फैक्ट यू शुड नो, अ सिंगल ह्यूमन क्रोमोसोम may have about million of nucleosomes. One chromosome have million of nucleosomes. Then now coming into chromosome. What is the structure of chromosome? So in this, firstly understand what is centromer. So a centromer is a point of attachment where two sister chromatids get attached to each other. मतलब दो क्रोमोसोम के पैर रहते हैं और वो एक पॉइंट से अटैच होके वन होता है, दो मिलके एक होते हैं, वो पॉइंट को क्या कहते हैं? सेंट्रोमियर। बराबर। देन क्रोमोसोम से क्या बनते हैं? जीन्स। क्रोमोसोम ट्रांसमिट होने पे जीन्स कहलाते हैं। सो व्हाट आर जीन्स? दे आर स्पेसिफिक सीक्वेंसेस ऑफ न्यूक्लियोटाइड्स on a chromosome that encode particular protein which express in form of some particular feature of body. So genes एक सीक्वेंस रहता है न्यूक्लियोटाइड पे एक क्रोमोसोम के ऊपर, उसे जीन्स कहते हैं। अभी आसान भाषा में क्या होगा? जीन्स आर part of chromosome only. So chromosome further is called as genes. On transmitting chromosome, they are called as genes. जीन्स एक कैरेक्टरिस्टिक है, कैरेक्टरिस्टिक ट्रेट है वो। अभी कमिंग ऑन टू सेल्स, then you may have often experienced this, that when you get a cut or a hurt, then another skins come up, right? So जरूरत क्या है न्यू सेल्स की? So पहला पॉइंट तो उसी में मिल गया, फॉर रिपेयर। वो कट हुआ, वो डैमेज हुआ ना, उसको रिपेयर किया। सेकंड पॉइंट है फॉर ग्रोथ। अभी बच्चे इतने होते हैं, बड़े होते हैं, बड़े 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 होते होते लंबे हो जाते हैं, तो क्या है? सेल्स ही ग्रो हो रहे हैं ना, फॉर ग्रोथ। देन फॉर रिप्लेसमेंट। नाउ सम सेल्स आर worn out or damaged, so through lysosomes they are taken off and new cells come on their place for replacement. Fourth, they are used for reproduction. In some unicellular organisms such as bacteria or fungi, they just get split into two parts called cell division. They just split into two parts and it is called for reproduction, sorry. Then cell cycle. What is cell cycle? Divide, grow and redivide. एक के दो हुए, दो के चार, और चार बाद में 8 हो गए। That is cell cycle. Then cell cycle has two phases, non-dividing and dividing. Coming on to now, this is the heart of chapter. This is the heart of chapter. अभी ये जो सेल डिवीजन की प्रोसेस है ना, वो कैसी होती है? वो है, सबसे पहला फेज रहता है इंटरफेस। इंटरफेस is divided into three phases. First, growth phase, that is also called as G1 phase. Second is synthesis phase, that is also called as S phase. Third is second growth phase, also called as G2 phase. So G1 phase में क्या होता है? RNA and proteins are synthesized, खत्म, और कुछ नहीं आगे। Synthesis में more DNA synthesized, chromosomes are duplicated. More DNA synthesized, chromosomes are duplicated. Done with S phase. Second growth phase मतलब G2 phase में क्या होता है? RNA and DNA are divided, or synthesized. Don't write term divide, write synthesized. Done with interphase. These all three phases together are called as interphase. Then cell division दो टाइप में होता है, माइटोसिस एंड मियोसिस। माइटोसिस is a production of diploid cells for growth and development. ग्रोथ एंड डेवलपमेंट के लिए माइटोसिस होता है और रिप्रोडक्शन के लिए मियोसिस. और क्या होता है मियोसिस में? हैप्लॉइड सेल्स. और माइटोसिस में डिप्लॉइड सेल्स. दे कैन आस्क यू दिस फॉर डिफरेंशिएट व्हाट इज माइटोसिस एंड मियोसिस के बीच का डिफरेंशिएट क्या है. Mitosis is used for growth and development and diploid cells, or you can also write that. So in differentiate, then you will write that in mitosis there are diploid cells, meiosis में haploid cells, mitosis for growth and development, meiosis for reproduction. इसके बाद there is a very important term called karyokinesis. Karyokinesis is division of nucleus. खत्म हो गई बात. साइटोकाइनेसिस भी है, उसका मतलब होता है डिवीजन ऑफ साइटोप्लाज्म. खत्म हो गई बात. फिनिश. बस. नाउ सेल डिवीजन में और 4 टाइप है. So now in cell division there are more 4 parts: prophase, metaphase, anaphase and telophase. So prophase में क्या होता है? The chromosome have short and thick and clearly visible inside the nucleus. Chromosome are visible. Centrosomes is splitting into 2 simultaneous duplication. मतलब क्रोमोसोम ना डुप्लीकेट होना शुरू करते हैं, ज्यादा कोई चेंजेस नहीं होते. न्यूक्लियर मेम्ब्रेन स्टार्ट्स डिसअपियरिंग. Duplicated chromosomes start moving towards equator. Metaphase, the sister chromatids move towards equator. The sister chromatids form an equatorial plane. And in anaphase, please keep in mind this way, that first will come prophase, then metaphase. So pro, meta, ana and telo. Pro, meta, ana, telo. This order is very important. They can ask you questions that identify the stage and tell and show with a diagrammatic sketch that which is the next phase. So keep in mind then what is significant in this stage, what are the observation they will ask you. So first observation, prophase, the nuclear membrane and nucleolus disappear. The duplicated chromosomes start moving, or the sister chromatids start moving towards the opposite poles, towards the equator. Metaphase, the sister chromatids get aligned in an equatorial plane. Then anaphase, the sister chromatids are get aligned towards their respective poles or opposite poles. Then in telophase, they get splitted. The chromosomes are split into 2 parts. Nucleoli reappear and spindle fibers disappear. That way. Then coming on to the differentiate between plants and animals. How does cell division is differentiate between plants and animals? So in animals, esters are formed, plants esters are not formed. Then cytokinesis by furrowing of cytoplasm, and in plants by cell plate formation. This is the important point, very much important in this chapter. Animals, cytokinesis by furrowing of cytoplasm. In plants, cytokinesis by cell plate formation. Occurs in most tissues throughout the body, occurs mainly at growing tips. Coming on to meiosis. What is meiosis? It is a reproduction cells or gametes. You should know only that much about it, not majorly. One more thing, that significance of mitosis: growth, repair, replacement and asexual reproduction. That means a unicellular reproduction. And fifth is maintain the same chromosome number in daughter cells. And what is significance of meiosis? Chromosomes are number is halved and there is remain same. Then mixing up of it provides for mixing up of genes. Maternal or paternal chromosome get mixed up. during the first division as they separate from homologous pairs. Now, here is the definition of crossing over. So what is definition of crossing over? So the exchange of chromated material, the exchange of genetic material from homologous pairs of chromosome is called crossing over. The exchange of genetic material from the homologous pairs of chromosome is called crossing over. And what is chiasma? It is the egg-shaped structure formed by crossing over between non-sister chromatids of paired homologous chromosome. It is a egg-shaped structure formed due to crossing over between non-sister chromatids and paired homologous chromosome. So coming on to the last point of the chapter, differentiate between mitosis, meiosis. First point you should keep in mind that where it occurs. Firstly, mitosis occurs in somatic or body cells, meiosis in reproductive cells. What is the reason for mitosis? It provides growth and replacement, repair, and meiosis for reproductive or gamete formation. When it occurs throughout the life, mitosis, growth, meiosis only in reproductively active age. Now number of daughter cells. Now parameters they might give, that is on basis of number of daughter cells. So you have to know that diploid means two daughter cells. Mitosis has two daughter cells. Haploid means four daughter cells. Meiosis four daughter cells. Number of chromosome. Full set of chromosome only half chromosome. Then number of nuclear division, single nuclear division, two nuclear divisions in meiosis. Then identify the chromosome identical and in meiosis randomly assorted. So according to me, the first 4 points are only, even for not four, first 3 points are only important. Yes. So with this, we end our chapter cell division over here, and hope you understood with the video. And if you liked the video, please do share with your friends, like, subscribe and press the bell icon so that you do not miss on any other notifications, as we are going to start with many new subjects and chapters, and we'll also start with the new chapters, and where I will teach you it in very fascinating way. So please do watch all the videos, and if you are new on a channel, please go and watch our previous videos about physics, English literature and geography. So till then, signing off for now. Thank you.